Thanks for the nice introduction. As in the introduction, if you guys have a question, just yell it out to me, and I'll repeat the question in the microphone so you don't have to worry about anybody hearing it. No? Okay. So what I'm going to tell you about today is a little bit about CRISPR technology and how we've applied that to the classroom, at least our initial application of it to the classroom. I think it does need a lot of work, and I think your input will be really important for that. Okay. So you guys have probably heard a lot about CRISPR in the news media, about how it could potentially change everything in terms of maybe potentially editing like the human germline and all sorts of other ethical concerns with it. But I think one thing that's really missed a lot is how fundamentally it's really changed the practice of biology, and how fundamentally it's changed research really. And so that's one of the things I'm going to try to get across in the talk that I'm giving. So just as an outline, so first I'm going to introduce CRISPR. This thing's a little anemic, but can you guys see that okay? So then I'm going to transition into genome editing and ethics. It would be interesting to hear your feedback on that. Some situations where I use CRISPR in the lab and really how CRISPR has changed the way that I do research. And finally, activities that we've tried with CRISPR in the classroom for master's students at SFSU, but potentially how you could adapt some of that to a high school classroom. Okay, so I want to start here with some fundamental processes just to make sure everyone's on the same page. I know you guys all understand this, but I want to make a few key points. Just as a fun note, after Bruce talked, I did have the title of this slide as Central Dogma of Biology, but I mentally deleted the dogma and turned it into fundamental processes. So already Bruce's talk has had a huge impact. So the point that I really want to make with this slide is that we have DNA, which contains all the information in the cell. That's then transcribed into RNA, which is then translated into proteins, which is really responsible for like our outward appearances and molecular interactions that occur in the cell. But the real key point is that if you change this DNA in any way, it's going to propagate through the RNA and then into the protein. So when we edit DNA, we're basically changing a lot about the outward characteristics of the organism potentially. Okay. So now I'm going to explain to you a little bit about CRISPR. So what is CRISPR? How did it come to be? How was it discovered? The answer is a little surprising. I think you guys all know about the concept of using it to edit genomes, but it really started out in a much lower key place than that. So in fact, it started out in one of my favorite journals here, the Journal of Bacteriology in 1987. So I always tell students in the lab, so I'm a postdoc now, so I get to dispense advice to graduate students, of course. And the thing that I always tell them when they have a question to me is like, well, have you checked 1980s issue of JBac? Because 100% of the time your answer will be in there. And of course, here's the answer in 1987 JBac, just for you all to know that I'm totally correct about that. Anyway, so there were some scientists in Japan who were just doing something very straightforward at the time, which was sequencing parts of the E. coli genome. So at the time, it was impossible to sequence the whole genome of the organism. There were just little snippets about on the order of a few thousand bases that you could sequence at any time. And they were sequencing the sequence of their favorite gene, which is IAP. I'm not going to discuss what that is. But next to the gene, they found this really strange thing. So they found this repeated sequence here, followed by some sequence that wasn't repeated. They weren't really sure what that sequence was. And then another repeat sequence, and then again another sequence that they didn't really know anything about. And this is actually really strange because in bacteria, they grow very fast and they evolve quickly. So having this kind of repeated DNA sequence in the genome was unexpected. Usually bacteria get rid of these repeated sequences. And then they looked at this and they were like, oh, well, we found this potentially cool thing, but we have no idea what the biology is. Okay? So fast forward a few years into the 2000s, my slide's a little cut off there. And we're getting more and more bacterial genomes. So now we're up to hundreds of bacterial genomes. And what some bioinformaticists noticed were actually that these repeated sequences were present in many of these bacteria. And so they came up with an acronym for these sequences, which is clustered, regularly interspaced, short palindromic repeats. And that's actually what CRISPR stands for. So it's kind of a mouthful. Nobody really says this out anymore, but that's what these sequences are, these repeated sequences. They are the CRISPR. Okay. So the bioinformaticians were really excited about these CRISPR sequences. They didn't really know what they were, but one of the key insights that they made actually was when they started to look at these spacer sequences, and what they noticed was that some of these spacers matched existing sequences in bacterial viruses or phage. And so they were thinking, wow, maybe these sequences have something to do with phage. And some people were even fanciful enough to say, well, what if this is some kind of defense mechanism against phage? What if this is somehow the bacteria is learning about the phage in the environment and then preventing the phage from attacking the bacteria? And this was all kind of a food for thought, if you will, until a very cool experiment in a very unexpected place. And that unexpected place was a yogurt company in Wisconsin. So this was just a few miles away, actually, from where I did graduate school. So it was CRISPR ground zero for a little bit. So one thing that I think not many people appreciate is that when you make yogurt at the scale that you need to feed hundreds of thousands of people across the nation, you have to make very, very large cultures of bacteria to help with the production of that yogurt. And one of the major issues that people see when they're working with these huge cultures is that bacterial viruses, these phage, will get into the culture and just decimate it. They will just destroy the whole bacterial culture, and then they have to start again from scratch and make everything up again. So this is a major economic issue for these yogurt companies, that these phage kill their bacteria. Okay? And then here's just a really awesome picture of phage actually injecting DNA into a bacteria. So basically the issue is that the phage come and inject their DNA into the bacteria, they replicate, and then the bacteria just explode. And then there are thousands and thousands more phage, which then go to infect the next bacteria. That bacteria explodes. So basically the whole culture has exploded in a very short order. Okay, so obviously the scientists at Danisco, the yogurt company, wanted to prevent this from happening, and they were doing some genetic experiments to try to understand this phenomenon they saw, which was when you treat a culture with phage, the vast, vast majority of the bacteria die. But there are some bacteria that don't die. They're resistant to the phage. Now, what makes those bacteria resistant? If they can figure that out, they can prevent their cultures from being taken out by the phage. And what they found, actually, was that when they treated the bacteria with this phage, the surviving colonies had incorporated a sequence from the phage into one of these CRISPR spacers. So this was the first best evidence that this is actually a phage defense mechanism, that this piece of DNA actually got incorporated into this CRISPR locus, and that basically they could take this new spacer sequence and then put it into a totally naive bacteria that had never seen the phage, and the bacteria would be totally resistant to that phage. So this was the best evidence that CRISPR was actually defeating bacterial viruses. So since then, we've learned a lot about CRISPR and how it works in defending against phages. So what I'm showing you here is a phage, and it's attached to a bacterial cell, which is what this beige thing is. So now the first thing the phage does, it attaches to the bacterial cell and then injects its genome into the bacteria. So the phage DNA is going into the bacteria. Now the bacteria doesn't want that, right? Because they will eventually explode and all their resources will be gone. They'll be dead. This is bad news. So in comes CRISPR. So again, here's this CRISPR array, this repeated thing with these spacers in it. And what happens is they're associated with these genes called Cas genes. That's what Cas stands for, CRISPR associated. And what these Cas genes do is they'll actually take a little piece of the phage genome, which is shown in this like red string here, and incorporate that into the CRISPR array, this red box. Okay, so now after the strain has done that, this CRISPR array will actually be transcribed into an RNA. Now this RNA is not, like I showed you in the central dogma, translated then into a protein. the RNA itself is actually functional. So the RNA does something here. So we have an RNA that contains all of these CRISPR arrays. It's then processed down into individual guides where each guide has a spacer. Okay? So now this particular guide is specific to a phage that this bacteria has seen before. Now again, notice that this red sequence is propagating through, so this guide RNA is based on this phage DNA sequence. Now finally, one of these Cas genes, the most famous one being Cas9, associates with this guide RNA. And the sequence of that guide RNA, remember, is the same sequence as the phage genome. So that RNA can actually deliver Cas9 to a piece of phage DNA. And once Cas9 is there, it actually cuts the phage DNA. And so once the genome of the phage is cut, it can't replicate anymore, and you're now resistant to that phage. Okay, so again, the phage DNA is degraded. In some systems, actually, the RNA is degraded. And I want to emphasize here something that I've kind of boxed out, which is that Cas9 is the most famous type of system, but there are other types of systems as well. Yes, question?
So how did that first step of, here's phage DNA, there's the red sequence from the phage DNA, its gene sequence, how did that get put into the CRISPR array?
Right, so the question is, how does this red sequence get put into the CRISPR array? And the answer to that is that there are other Cas genes that I didn't discuss that actually will associate with this phage DNA, cut a piece out, and then insert it into this CRISPR array. And it's not any particular piece of DNA, in the case of the type 2 CRISPR system, any piece of DNA along the phage that works for interference or any piece of DNA will work for interference, basically, cutting the phage.
Why don't those enzymes attack the bacterial chromosomal plasmids?
Mm-hmm. Right, so why don't they, so they do attack plasmids actually, so it's not specific to phage, but any foreign piece of DNA, as we describe these things, coming into the bacterial cell could get incorporated into a CRISPR array. Now, if you think about it, what happens if you put a piece of the bacterial genome into the CRISPR array, right? It'll go through this whole process of making the guide RNA, which will associate with Cas9, cut the bacterial genome, and the bacteria will die instead of the phage. So that's selected against, basically.
This we used to set CRISPR kind of loose and wild, but put in some spacer sequences for some of the more common sequences found in antibiotic resistance.
So the question was, a lot of antibiotic resistance is carried on plasmids. Can we use CRISPR to target those plasmids and destroy them? That's one of the potential uses for CRISPR, yes. I think people are looking into that.
Were they using this for the yogurt you were talking about?
Is this the system described in the yogurt? So it's in the yogurt system, there's actually a different type of CRISPR system called a type 1 CRISPR system. But the concept is very similar. A piece of the viral genome goes into the CRISPR array, Cas proteins go and then cut that. It's a slightly different set of proteins though.
But it is a foreign piece of DNA that's being inserted. So
yeah.
Then is our yogurt genetically modified?
So that's a big question, right? Like, is the use of CRISPR technology count as a genetic modification, or does it count as, sort of, the right word is kind of domesticating the way you domesticate a crop? And that's a significant regulatory question at the moment for which there is not a clear answer, I think.
Can you go over again, going from the gRNA to the Cas9 part?
Yes. So the guide RNA actually has sequences on it that bind directly to Cas9. So there's an affinity for the guide RNA for Cas9 directly. Yeah. Okay. With that, I'd like to show a fun video for how Cas9 actually recognizes viral DNA that was made by a colleague of mine, Cy Redding, in part. And this could be a fun one to show your class, for instance. So this is about how Cas9 finds the target DNA.
When bacteria become infected with a virus, they recruit a protein to hunt down and cut its DNA. That protein is Cas9. It looks something like this. Bound to Cas9 is a strand of RNA. It's the key to finding the viral DNA. a 20-letter sequence stolen from the virus when it last infected. Cas9 must search millions of base pairs to find the matching sequence in the viral DNA. Instead of looking directly for the entire code, it first recognizes an incumbent 3-letter sequence called PAM. There are thousands of PAM sites, but only one exists next to the matching sequence. Cas9 unzips the DNA at each PAM site and checks to see if the rest of the sequence is complementary to its RNA. If it doesn't match, Cas9 eventually falls off and the search continues. When the matching sequence is finally found, Cas9 knows it's locating the viral DNA and cuts both strands. Without its DNA, the virus can't replicate. Not today, punk, and not tomorrow either, thanks to Cas9 and its guiding RNA.
Okay,
That's great.
so that was a lot of fun, but don't applaud me, it's applaud the group there, fantastic video. Does anyone have any questions about this process? Yeah.
When the gRNA is, or when the job's done, the gRNA just release, kind of like a tRNA once it's relieved, once the
Yeah, so the question is whether or not the guide RNA actually is released from Cas9 after cutting. And I think that there's not a clear answer, but it seems likely that the guide RNA is just retained unless there's another guide RNA that competes with it. So if it was the only guide RNA in the cell, it would probably just remain bound to Cas9. If there's a competitor RNA, it might switch between those. It definitely does. Yeah. Great question. Okay. So I know what you're thinking now, which is how did we get from phage biology to genome editing? And it's actually very simple. So the key thing in editing a genome, the most important thing, is actually to get a DNA double-strand break. Because as soon as you get a DNA double-strand break, you can then insert any DNA you want essentially into that site of breakage. So Cas9 offers a way of getting that double-strand break. So again, we have a stretch of DNA here. We have our molecule Cas9 and a guide RNA. So the guide RNA is complementary to the DNA. And Cas9, what it has is two active sites that act like scissors. So these scissors will cut either strand of the DNA, and that will create what we call a double-strand break, which is basically when both of the backbones of both strands of the DNA have been cut. So, so you can think about it kind of like a guided molecular scissors that will cut any particular sequence depending on the sequence that is specified in the 20 nucleotides. And as you might imagine, from a biotechnology standpoint, it's very easy to change these 20 nucleotide sequences. Okay? So the question is, once you have a double strand break, what happens? How can we actually use this to do genome editing? Well, there's kind of what I would consider a passive way and an active way. So if you just cut double-stranded DNA in a eukaryotic genome, now we're talking about eukaryotes and not bacteria, what happens frequently is you get a process called non-homologous end joining, which you can totally forget about immediately. But it's the acronym in the name is not important, but the important thing is that there's no template to fix this error in this case, right? So it's kind of like, I don't know if you've ever had this experience where you've taken something apart and you're like, I'm gonna fix this thing, right? It's broken, I'm gonna fix it. Take it apart, and then you're like, all right, what did it look like? I don't know. So, and then you're like, well, I mean, I don't really remember what it looked like, so maybe if I can just like stick these things together, kinda, and then like duct tape it, like that'll be good enough, and then sometimes it is good enough, right? So, basically that's what the cell has done here. It's stuck these two ends together, and a polymerase basically comes by and fills in a few nucleotides in this situation. So now the sequence is different than the original sequence by a few nucleotides. Yes?
It does the blunt end cutting. It never does the staggered end ever.
So with the question is whether or not Cas9 does a blunt end or a staggered cut. And Cas9 always does a blunt cut. There are other editing enzymes out there, such as CPF1, which is another CRISPR enzyme that's really functionally related to Cas9, but it actually cuts staggered, more like a restriction enzyme would cut. It's a great question. So yeah, Cas9 always does a blunt cut. Okay, so now let's think back a little bit to the central dogma that I showed you at the very beginning. So what effect do you think you would get from inserting just like a few nucleotides into this gene? Right, so this is going to be transcribed to an RNA, that RNA is going to be different. It's going to be translated into a protein. That protein would be different. But one thing to think about, right, is that because the DNA code is a triplet code, right, if you insert one or two nucleotides in here, now your code is totally off. And what that usually does is it results in a premature stop codon. So you have a translational stop codon. And that protein that usually comes out of that is now truncated. And so it typically doesn't work. So the typical outcome of this sort of duct tape repair mechanism is that the gene is inactivated. So just by passively cutting genes in eukaryotic genomes, you can inactivate them using this kind of methodology. And so this has been really awesome for doing like very high throughput screens of eukaryotic genomes where we're just cutting thousands and thousands of genes and looking to see what happens, because when we cut the genes, most of the time they're inactivated. Okay. Yes?
So this leads to a frameshift mutation and inactivates genes. So basically you want to inactivate genes.
So it typically results in a frameshift mutation. Frameshift mutations typically inactivate genes. If it doesn't result in a frameshift mutation, it will often result in a missense mutation. So there'll be other amino acids here that are potentially deleterious to the protein. It's a little less clear whether or not the missense mutation will definitely give you a loss of function of the protein. Frameshifts typically do give you loss of function of the protein because it just cuts off a huge part of the protein, and that's no longer functional in that case. Okay, great question. But this is some people would argue that this isn't truly gene editing, right? It's definitely disrupting a gene, but it's not really like replacing it with something that we're interested in. So that's the other possibility. So let's say instead of just breaking a gene, I want to put this awesome thing into the genome. Now, it doesn't actually matter what awesome thing you want to put in there, right? This awesome thing can encode any DNA sequence. Let's say you want to put a green fluorescent protein in the genome of a eukaryote. That's fine. You can do that. You can put it more or less wherever you want, as long as you don't disrupt something really important. And the way you do that actually is by flanking your awesome thing with DNA that looks just like the DNA that you originally cut. So you have an awesome thing in the middle, and on the ends you have pieces of DNA that look like this DNA. And basically what happens is, through a process known as homologous recombination, this awesome thing gets stuck into the genome. So basically in this case, remember for the non-homologous end joining situation, I was just like, well, I have these two things and I don't remember what they look like, so I'm going to duct tape them together. For homologous recombination, we're just like, oh, I have these two things. Oh, hey, there's a thing that looks like that thing. This is definitely how it's supposed to be. I'll take that thing and put it in here. Okay, great, now I fixed the problem. But in the process, you've incorporated this awesome thing into the genome because the cell recognizes these ends, basically, and not the awesome thing directly. So now you can put any awesome thing you want into the genome just by doing a double-strand break and then having this, what we call, editing template. And these editing templates are actually very easy to come by these days because of DNA synthesis technology has improved so much that it's very easy just to order hundreds of nucleotide pieces of DNA and simply insert them into the genome. So you can do this at a pretty fast clip. Okay. All right. Yes, questions?
So it's still pretty non-specific, right? Because how do you get the editing template to recombinate like right into that spot? 'Cause
Yes.
Isn't that one like way harder than
No.
AH?
That's a good question. So how does the editing template know to get into this spot? And maybe it's because my slide's a little bit not great here. But the reason this works is because this sequence here is exactly the same sequence as this sequence here.
But
So
just like
that's-
the spatial problem, like do you fill the cell just with tons of editing template and they just Brownian motion just get in the right
Yeah,
spot?
that's a great question. So the question is, like, how much editing template do you really need to get this to work, because it's got to find in this DNA. So yeah, you would use a considerable amount of template in order to make sure that you have enough in there that the repair enzymes can recognize it and put it in there. But keep in mind that this is a really bad situation for the cell. Having this double strand break, if it's not fixed, will cause the cell to die. So the cell, I hate to anthropomorphize too much, but it's like the cell really needs to fix this. So as soon as it detects that this has happened, it's upregulating its repair machinery, and that's driving all these processes like combining DNA together. So it's a little bit less like passive diffusion and a little bit more like actively trying to fix this thing. But yes, you do need a lot of DNA to work efficiently.
What was the model of organism which was used here? Was it plant, animal, bacteria?
Yeah, so the question was, what kind of model organism, what kind of DNA am I cutting here? Is it plant, animal, or bacteria? So this actually works very well in plants and animals. Any particular system that's been tested, I think, it works very well in. So any kind of eukaryotic cell, including mammalian cells, this process works, human cells works in. It doesn't actually work very well in bacteria, and the reason for that is bacteria don't have non-homologous enjoining. Most of them don't. Some of them do, but when they do have it, they often express it in situations that are not conducive to doing this gene editing. Because bacteria most of the time actually have multiple copies of the chromosome because they're dividing so quickly, and they just use that as a template. Because if you think about it, right, using a template for putting something together is better than duct taping it. So that's the preferred mechanism of bacteria. Okay, any more questions on this? Because I think this is an important slide.
Your awesome thing does necessarily have some sort of molecular marker. Is there another way to identify other than, like, say, using PCR and going that route?
So the question is there any way to identify this awesome thing other than selection? So I think the answer is yes. The efficiency is such that you can get some things without selection directly for this element going in, because this in a way it's not truly a selection, but it's driving this recombination process basically by recruiting these repair enzymes. So yes, I think you can do this without selection. Now you're looking at efficiencies, I think, below 10%, maybe below 5%, so you're definitely going to have to hunt for things. I think typically when people do these things, they're putting at least some sort of indicator in there, whether or not it's a directly selectable marker, like an antibiotic resistance marker, or a fluorescent protein, so that they can just tell whether or not something's there and being expressed, just in practical terms.
How do you get the Cas9 CRISPR protein and the template into a eukaryotic cell, into the nucleus?
Yeah, so the question is, how do you get this stuff into eukaryotic cell and how do you get it into the nucleus? So what Feng Zhang's lab developed early on in this editing process is actually they stuck nuclear localization signals on both ends of the Cas9 protein. So once that protein is in a eukaryotic cell and expressed, it will be delivered to the nucleus. Usually I think people, when they do this sort of editing in eukaryotes, they have plasmids that they can electroporate into the cells to express it. Alternately, you can actually have purified Cas9 and purified guide RNA complexes that you can directly electroporate. I think there might be differences in efficiency for those sorts of things. But I don't actually have direct experience with that. Remember that I'm a bacteriologist. More questions?
Is the benefit to doing CRISPR versus a traditional restriction enzyme cloning that a PAM site is more readily available than perhaps a restriction enzyme site?
So thinking about this, and the question is basically how do we compare this to restriction cloning? And the cool thing about this is that, I think Yeah, this can all happen in a cell, right? So with restriction cloning, you're basically assembling the molecule in vitro and then transforming it into the cell. This is happening in a cell in the chromosome of that organism. So this is a heritable change that's happening to the DNA in that organism. It's not like an extra chromosomal element that you're putting into the organism that potentially could be lost or is not actually part of the genome. I think that's a really important distinction.
How many cells or what type of cells do you use this in? Like, if you whatever animal model you're using.
So as far as types of cells are concerned, so the question is what type of cells can you do genome editing in? And I think people have done genome editing in essentially all types of cells. All types of cells that have been tested, it's worked in. As far as cell numbers are concerned, I don't actually have the right expertise, I think, to answer that question directly. And actually, I should mention that I don't know if my email is anywhere, but if you email me these questions, I can definitely ask my colleagues that do CRISPR in eukaryotic cells to give me some answers for that.
With this awesome thing, does it have to have some sort of promoter or come with its regulatory region as well?
It doesn't necessarily, right? So the question is whether or not you have to separately express this awesome thing. I think the thing is like this awesome thing could be a protein that you express, and in that case it could have a promoter and whatever, or it could just be change to the coding sequence of the gene that you're interested in, right? In which case that gene would have its own promoter and everything, right? So any number of possibilities are possible with this technology.
Is there success variation depending on whether the gene's located in neuchromatin or heterochromatin? You think that if things are supercoiled or all silent?
So the question is whether or not the chromatin state matters for CRISPR editing, and I think there is an effect on that. So the more tightly packed DNA is harder to access by the Cas9 protein, but it still works. That's my impression.
What's the limit on how big your awesome thing can be?
What's the limit on the size of the awesome thing? That's a great question. I don't necessarily know the answer to, but I know that people are putting many kbs worth of DNA into eukaryotic genomes. The actual upper limit in terms of size, I don't know, and it's probably changing by the day.